Welcome back once again. Uh, in the chapter The Molecular Basis of the Inheritance, we will continue RNA world, what we call as RNA world. Okay. From foregoing discussion, an immediate question becomes evident: which is the best genetic material? It shall be discussed in details in the chapter on the chemical evolution, but briefly we shall highlight some of the facts and points. Okay. The RNA was the first genetic material, and there is now enough evidence to suggested that essential life processes such as metabolism, translation, splicing, etc. evolve around RNA. RNA used to act as a genetic material as well as a catalyst. There are some important biochemical reaction in living system that are catalyzed by RNA catalyzed and not by protein enzymes, but RNA being RNA being catalyst was reactive and hence unstable. Okay. Therefore, DNA has evolved from the RNA, which chemical modifications that make it more stable. DNA being stranded and having complementary strand further resist changes by the evolving process of repair. Yeah, we can see this is the this figure given by Watson Crick mode of the semi-conservative DNA replication. What a fantastic. Okay. GC 8080 GC 8080 GC 80 GC 80C GC CG 80 80 80 AC, and yeah, we can see this the following steps. It will be continue. Next one is replication. The subtopic is in this regarding. While proposing the double helical structure for DNA, Watson and Crick had immediately proposed a scheme for replication of DNA to quote their original L statement that is as follows: it it has not escaped our notice that a specific pairing we have postulated immediately suggested a possible copying mechanism for the genetic material. Okay, the scheme suggested that the two strands would separate and act as a template for the synthesis of a new complementary strands after the completion of replication. Each DNA molecule would have one parental and one newly synthesized strand. Thus, a scheme was termed as semi-conservative DNA replication. Is it? Now, the experimental proof we will see. It is now proven that the DNA replicates semi-conservatively. It was shown first in Escherichia Escherichia coli, what we call as Escherichia coli, and subsequently in higher organisms such as plants and human cells. Matthew Michelon and Franklin Sethel performed the following experiment in 1958. They grew equally in medium containing NH. This is 15. Okay, Cl. 15N is the heavy isotope of nitrogen. So important thing we can see of nitrogen as the only nitrogen source for many generations. The result was that N15, okay, 15N sorry, was incorporated into newly synthesized DNA as well as other nitrogen containing compounds. These heavy DNA molecule could be distinguished from normal DNA by the centrifugation in the cesium chloride CSCl density gradient. Okay, please note that N15 is not a radioactive isotopes. Okay, it is not radioactive isotope. It can be separated from the N14 only based on the densities.